Good morning, everyone. My name is Zara Sheikh, and today I will be giving a brief talk on transport of local anesthetic lidocaine across pharyngeal air-liquid interface cell model. I am an early career researcher in the Respiratory Technology Group at the Woolcock Institute of Medical Research based in Sydney, Australia, and I recently finished my PhD from University of Sydney under the supervision of Professor Daniela Trainee, Dr. Huizing Ong, and Professor Paul Young. Let me give a brief background about our research. Lung epithelial cell line has been shown to be promising in vitro models of airway epithelia as it simulates the airway physiology in vivo. Airway epithelia is the principal barrier to drug absorption, and this is the prime reason why human lung epithelial cell lines are widely studied. Our lab has developed in vitro models of human lung epithelial cell lines, namely CALO3, CUFI1, and NULI1, and these in vitro models have found numerous pharmaceutical applications in terms of studying the mechanisms of drug transport, studying aerosol deposition, control release studies, as well as identification of possible drug-drug interactions. Most of the research utilizing these human lung epithelial cell lines is directed towards the lower respiratory tract, and the nasal cavity has also been widely explored. However, the oropharyngeal region has been overlooked. There are a number of therapies targeted for the throat, namely anesthetics, antiseptics, analgesics, as well as immunosuppressants. Tetrad 560 is a human pharyngeal cell line that is widely used to study immunological pathways. However, an in vitro model with optimized culture conditions is not yet developed, and whether these in vitro models could be used to study drug transport has not yet been explored. Considering the number of therapies that are targeted for the throat, there is an imperative need to develop such an in vitro model to study aerosol deposition targeted at the throat. And therefore, our study aimed to develop an in vitro air-liquid interface model, in short ALI model, of the Tetrad 560 cell line, and characterize the model in terms of its differentiating characteristics such as paracellular permeability, formation of tight junctions, mucus secretion, and cytokine production in response to an inflammatory stimulus. And finally, we investigated whether the developed model could be used to study drug transport using a local anesthetic throat spray, lidocaine. An air-liquid interface model is established by seating the cells at the apical layer of a semi-permeable support membrane and placing media at the basolateral chamber. After 24 hours of seeding, the media is removed from the apical layer and the cells are allowed to grow at the air-exposed surface. The cells multiply and grow and differentiate throughout the ALI culture period, and at the end of the ALI culture period, they show the differentiating characteristics in terms of its mucus production and formation of tight junctions that enables passage of small molecules, ions, solutes through the gaps in between the cells, thus facilitating paracellular transport and showing that epithelial barrier integrity has been attained. There are a number of methods to characterize this air-liquid interface model in terms of its mucus secretion, cell viability, formation of tight junctions that enables paracellular transport, and looking into the tight junction proteins through immunofluorescence. The matured ALI model is then studied to see whether it produces cytokines in response to inflammatory stimulus such as LPS or lipopolysaccharide, which is a bacterial component that stimulates inflammation within the lung. These models are also stimulated with the viral component known as polyic to see whether these models can be used to study viral infection. And also this ALI model is explored to see whether it can be used to study drug transport. To optimize the seeding density of our ALI model, we have used 3 seeding densities. The light gray bars represent 30,000 cells per well, the dark gray bars represent 60,000 cells per well, and the black bars represent 80,000 cells per well, which have been used throughout our study. Transepithelial electrical measurements, in short TEER, is an indirect measurement of formation of tight junctions and shows epithelial barrier integrity attainment. An increase in TER values shows that tight junctions have formed. TER measurements were performed every week for 21 days, and we saw a significant increase in TER values from day 7 till day 18, and this shows progressive tight junction formation till day 18 of ALI culture. for all the three seeding densities. No significant differences were found between day 18 and day 21, suggesting that tight junction formation has taken place at day 18 of ALI culture period. These results were further confirmed by sodium fluorescent permeability assay. Sodium fluorescent is a paracellular marker that indicates whether tight junctions have formed by a decrease in the apparent permeability value or PAP value. The PAP value decreased significantly at day 18 for the intermediate and high seeding density compared to day 7 and day 14, and then plateaued till day 21, again confirming that tight junction formation has taken place at day 18 of ALI. For the lower seeding density, the PAP value decreased at day 21, showing a longer time for the tight junctions to form, and therefore for our subsequent experiments, we used 60,000 cells per well and 80,000 cells per well. The cells were stained with calcium AM to see whether cells are alive at day 18 of ALI and compared with day 7 ALI models, and we saw sufficient number of live cells at day 18 of ALI. The cells were stained with Alcin blue. Alcin blue is a surrogate marker of mucus production as it stains the glycoproteins present within the mucus, and increase in blue intensity indicates mucus production. Sufficient mucus production was observed at day 18 of ALI for both the seeding densities, thus showing that these ALI models are capable of differentiating and producing mucus. Next, the ALI models were stimulated with LPS and poly-IC. LPS is the bacterial component that stimulates inflammation within the lungs. LPS-stimulated cells showed significant increase in production of the pro-inflammatory cytokine interleukin-IL-8 compared to the unstimulated cells that served as controls after 24 and 48 hours of stimulation for the intermediate seeding density of 60,000 cells per well. Similar results were obtained for the higher seeding density as well, showing significant increase in production of the pro-inflammatory cytokine IL-8 in the LPS-stimulated cells compared to control, overall showing that this ALI model can be used to study bacterial infection. When the cells were stimulated with the viral component poly-IC, the stimulated cells also showed significant increase in the production of the pro-inflammatory cytokine IL-8 compared to control for both the seeding densities, again showing that this model can be used to study viral infection. Finally, the ALI models were investigated to see whether it can be used to study drug transport. Lidocaine solution was added at the apical layer. Basolateral samples were collected over a 4-hour period, and we observed that cumulative mass of lidocaine transported across the ALI cultures increased with time, with no significant differences in the cumulative mass of lidocaine transported between the two seeding densities. Approximately 50% of the drug deposited in the apical layer was transported across the ALI cultures, and notably no drug was found inside the cells represented as cellular, suggesting that lidocaine may have transported via the tight junctions in between the cells, thus enabling paracellular transport. However, the paracellular route for lidocaine must be further explored. To determine whether lidocaine deposition altered the epithelial barrier integrity of the cells, TER measurements were performed before and after the experiment, and we saw no significant differences in the TER values before and after the experiment, showing that epithelial barrier integrity was maintained. Furthermore, the apparent permeability values of the paracellular marker Fluna for untreated cells and treated cells post-transport was found to be very similar, with no significant differences between them, again confirming that epithelial barrier integrity was not altered by lidocaine deposition. Overall, our study showed that our developed ALI models of the pharyngeal cell line Detroit 562 could be used to study drug transport. To summarize our results, 60,000 cells per well at day 18 is the optimum culture condition for Detroit 562 ALI model, and currently we are developing our novel 3D printed throat model incorporating our developed cellular model to simulate aerosol deposition in the oropharyngeal region and study therapies targeted at throat. I would like to thank the entire Aesthetic team and my supervisors and the DDL organizing committee for giving me an opportunity to present my research. Thank you.